to all of our speakers and welcome back everyone to the main room. We will now deep dive with two of our invited speakers in a fireside chat. Please allow me to introduce the moderator of the session, Dr. Paul Beaumont, Principal Data Scientist at QuantumBlack. Thank you all, and over to you, Paul.
Thank you, Tran. Good evening, everybody. It's my great pleasure to be able to welcome three great panelists to our fireside chat this evening. Firstly, we have Dr. John Huang, Principal Investigator and Biostatistics Platform Lead at Singapore's Institute for Clinical Sciences, A*STAR, and also Adjunct Assistant Professor at Duke NUS Medical School. We have Dr. Nam Sivo, Director at the Biomedical Informatics Center at Vingroup's Institute of Big Data. And we also have Nguyen, Chief Technology Officer, also at Vingroup's Institute of Big Data. Welcome all. Thank you all for joining us this evening, and thank you, John and Sivo, for your great invited talks as well. In this session, we'll be taking a selection of questions from the audience. So anyone in the audience, please do feel free to use the Q&A functionality to join in with the discussion. But maybe, just to start things off, I can first ask. So Sivo and John, you're both involved in cohort studies, so the 1,000 Vietnamese Genomes Project and the Human Development Program, respectively. For anyone who wasn't able to catch your talks, because as we were discussing before, unfortunately you were on at the same time, please could you maybe just introduce the work that you've been doing, and maybe it would also be great if you could help us understand in these projects just how important is ensuring that your work reflects the causal relationships.
Yeah. Maybe I first or John first?
Sure, please, go ahead.
Okay, so like Paul mentioned, so in my slide, I already talked about the 1,000 Vietnamese Genome project. So overall, this project is our effort to reduce the bias in the data between European sample and the other population sample. Here, in particular in Vietnam. So before that, Vietnam has little data, genomic data. So the 1,000 Vietnamese Genome Project is the first effort to create a large-scale genomic data for Vietnamese population. So for that project, we aim several things. The first, we try to do the whole genome sequencing of more than 1,000 Vietnamese people, just healthy people, so that we can have some a better understanding of genetic structure of Vietnamese based on a large-scale cohort. And the second thing, we try to do genotyping and sequencing of the patient, the Vietnamese patient, like type 2 diabetes, and some cardiovascular, and some patient with adverse drug reaction, ADR. So that based on that, we can have better understanding of the PGX pharmacogenomics of Vietnamese patients, like the ADR in SCAR and NSAID hypersensitivity and DLE. So that based on that, we can develop some application that we can lead to genomic medicine implementation in Vietnam. So for the 1,000 Vietnamese Genome Project, so it's kind of similar to 1,000 Genome Project before, just 20 years ago, maybe 10 years ago. But the thing is here we focus on only one ethnic group, is the Vietnamese, and even one major ethnic group in Vietnam is the Kinh. Kinh is a major ethnic group in Vietnam. And the second thing, we aim to use and we aim to use the genetic data and phenotypic data from Vietnam in the best way. Means that 1,000 Genome Project, they don't obtain the phenotypic data, but here we obtain as much as possible the phenotypic data, like the BMI, the height, the weight, and some other information. So that is very important for medicine, genomic medicine in Vietnam. And that is some important information about 1,000 Vietnamese Genome Project. And the second question from Paul is causal model here. I think it's very important because in addition to the 1,000 healthy people in Vietnam, we are sequencing and genotyping around more than 4,000 samples from patients. And for those patients, we want to get some novel variant that can use to predict like an idea adverse direct reaction, or can predict the type 2 diabetes disease, so the risk. And based on that, we want to see some specific variant that can cause the disease. And for that, so we are developing both. We develop some model to improve the prediction accuracy, but both we want to get some some insights, some biological insights of why some patients they get idea and some others not, and why some patients they get type 2 diabetes and some others not. So that's causal model is very important there. So that we should also we are trying to balance between the accuracy of the model we want to predict in the best way, but also we want to try to find some specific hotspot variant there to explain some behavior of the patient. Yeah, that's our project.
Awesome, thank you. I think later on we might probably get into some of these trade-offs as well around kind of accuracy versus kind of causality potentially as well. So let's see where the discussion goes. But no, awesome, thank you. And John, maybe would you be able to help share a little bit on the Human Development Program as well, please?
Yeah, sure. Thank you, Paul, for having me. I'm very privileged to be alongside these two other distinguished panelists. So in terms of what my talk briefly overviewed was some of my experiences working to consult on some of the clinician scientist projects. So in general, our institute is interested in early child health and human development, and that also encompasses maternal and family health. And I would say that, so I'll say briefly that I sort of covered two points that were specifically related to causal inference in my talk. The first one being, how do we deal with unmeasured confounding in studies of non-randomized treatment, so not in studies that aren't RCTs. And it's a major challenge when we're studying health, because oftentimes we'd like to run RCT, but we can't. And for many different reasons. So I was talking specifically in the context of looking at long-term effects of assisted reproduction and whether or not we can use molecular data to help inform that. And I think just dovetailing a little bit with what Zibo was talking about, I think they're complementary efforts to understand the causal relationship between genes and gene networks on biological function and therefore disease. But then also, how do those gene networks and expression and everything fall within sort of the general framework of our daily lives, of things that we can modify, whether it's through diet or through medicines. And so they do require, some of the similar types of approaches in terms of causal inference, but also some unique ones as well. In terms of trying to understand modifiable molecular and genetic features is quite apart actually from understanding sort of the basic genetic function, but they're sort of complementary. The other part that I'll only touch on about is, how we use, how do we incorporate machine learning into causal inference? And there's obviously been a lot of work on this, but I think where sort of the rubber hits the road is the major challenge. And this is something that, was raised by Professor Kripson in the keynote speech leading off this session, which was, it's so critical, right, to be able to get domain expertise in order to help form your DAGs and help form an identifiable and estimable model. But I think what we run into practically, as analysts, is that oftentimes that's very challenging. In terms of the interface between a domain expert and the person doing the analysis, it's hard to ask the right questions. It's hard to sort of focus in on, what is it that they, that they actually want to estimate, and can they actually, it's nice to say, yes, get rid of that node, but most of the time, and I wouldn't either want to get rid of any nodes, right? Because it's always a possibility that something can, but anyway, aside from that, there's also technical issues that we need to be concerned with when we're trying to use machine learning to estimate causal effects. so that's what I talked about in my very brief talk. But I think to answer your question a little bit more, which is I spend a lot of time trying to help elicit from these clinician scientists or the other basic scientists and other researchers whether or not the question that they're interested in is actually in a causal estimation type of question. And that's different than, I think, just sort of broadly understanding cause or broadly causal inference, because at the end of the day, we hope everything that we do is causal, right? Like even if I'm building a prediction model, I hope that it will influence practice, and I hope that practice will be improved by that. So in and of itself, there's something causal there, but I think it's a separate set of questions than the question of, can we, given data that we have some better understanding about how the world works, and specifically how the world might change if we were to modify or manipulate some part of it, right? So that kind of discussion is always ongoing in the types of work that we do.
Yeah, no, cool. Thank you. Totally agree. And you sparked a thought in something you just said, that we're always hoping that whatever we're doing is going to end up being causal. Like these predict then optimize paradigm of like the way of using machine learning models sometimes inherently is sort of assuming that these models, which are quite often never causal models, are in some way causal. I think maybe that segues on quite nicely into a follow-up question. So I think kind of, as we know, generally, sadly, most machine learning models applied straight out of the box will care not for causal relationships at all. So kind of, do you think that this is ever going to be okay in other types of work outside of maybe kind of the work that you do? But does thinking causally in machine learning have to be a big overhead? Or do you think that this can actually be married together?
I'm trying to understand the question a little bit better. Is machine learning in conflict with causal inference, or what?
I'd hate to be quite so controversial, but but in terms of like, so do you think building any sort of machine learning model without kind of thinking about the causal aspects is ever okay? Or, and I guess it is a slightly different question, sorry for lumping it together, but kind of, I think one of the reasons that I get a sense that some people would avoid trying to build a causal model is they think that it's quite a large overhead to doing it. Do you think that always has to be the case?
Yeah. I think so. I mean, to answer the easiest question, no. I mean, obviously, machine learning models do not have to be explicitly causal. They do not have to consider, let's say, causal diagrams or how these things fit into causation. I mean, certainly, okay, so take out a lot of the machine learning that's done in, let's say, A/B testing, because that's just explicitly causal and treatment related. But let's just say using machine learning for prediction modeling or for classification, those sorts of things. I mean, where the causation comes in, I think we referred to it before, is in build whatever model you want, then test it, right? So the causation happens at that point in time, is when you implement it, you put it up against a competitor, you put it up against best practices. Does it improve outcomes, right? There's a lot of different theory that can go into what you think that best model is, and you can have constant conflicts over it, but then the actual test of it is later on. So, I mean, generally speaking, it doesn't have to be in conflict if you think about it in that sort of realm.
The proof is in the pudding, sort of thing, at the end of the day, and how you use the model. Okay, awesome, thank you. Maybe I can ask you a bit of a question around the data side. So many people, particularly outside of the healthcare, but also certainly inside the healthcare domain as well. So for instance, like phase 4 trials, non-RCT trials, development studies as well, are only able to work with observational data. They never get to work with RCTs. I know kind of we've talked around them a little bit, but kind of, do you have any particular best practices that you would normally follow to ensure that your analysis on observational data can be as causal as possible? I'm just intrigued to see, do we share sort of techniques, or do you see different techniques being most appropriate?
Oh, sorry. So I am just muted. So I am interested in it as well because you think that I am interested in it is because of the data in Vietnam. Sometimes we can generate the data based on some careful study, some careful design study. But sometimes, especially in Vietnam, we can get data from some other means that we don't have some choices. Yeah, for example, the data from some other hospital, the data from some study before, but they don't design for some objectives that we want. I don't have any specific, basic question. I don't have any specific practice or practice analysis to answer the question just as causal as possible. But I think based on my experience, based on some study here, we have some machine learning model that we can apply to some the whole data that we have, a whole bunch of data, big data. So we are developing some big platform here, the platform for big data here. And for just platform, we carefully design some model, like let's say the graph model that we define previously, some relationship between some entity, biological entity, and the phenotype data and genotype data and some other. And based on some very careful design model like that, we try to get the data submitted to our platform in some structured way. So we put that into the user, that people submit the data into our platform, and based on some models that we already designed, that when people submit it, people will be structure their data a little bit, and then we can, based on those model, those let's say the graph model, we can infer some other relationship. And just think that one of the things that we are doing here to get the data more structure and to get it maybe, if I understand correctly, as causal as possible in some way. So that's my some short thought.
Maybe I can add a quick comment to that. So most of the time, a randomized critical trial is pretty expensive, but it provides the most objective information. But when you have a carefully curated data with good metadata and phenotype data, and select those data based on the real distribution, like good in the real world, that can provide a higher quality for the AI model inference. So that really reinforces the point that Dr. Nam mentioned earlier, that good high quality phenotype data, especially in clinical settings, is really important in good AI healthcare model.
John, is there anything that you want to, any best practices that you do with your observational data side?
Yeah, the thing that I think has been very motivating for people to get everyone on the same page is the target trial paradigm. And I don't know if people are familiar with it, but it was proposed, I don't know, maybe 5-10 years ago, where you say, I have my observational data. If I were to have done a trial on this, how would I have structured this data? What would be the inclusion criteria? What would be the time zero? What would be the induction period, the treatment, the treatment dosage or period? These kinds of considerations, then analyze it in that way. And so you can bring in machine learning tools, you can look at propensity scores and that kind of thing. But the real sort of magic of it is in the setup, is in the design, saying this is who I would include, this is the kind of exposures that I would consider to have, for example, consistent effect. Right, and a lot of the things that we deal with are is we suppose or we think that there's treatment effect heterogeneity. Certainly, that's one of the things we want to look at. For example, with pharmacogenomics, we want to know if there's treatment effect heterogeneity. And so those are the kinds of questions you can ask if you structure your data in a certain way, so that I say, okay, this is the kind of, maybe I would consider a factorial trial or something like that. And then you analyze it in that way. And I think that helps really get a lot of people on board as to what exactly you're doing. You talk about causal inference, you're like, okay, yeah, everybody wants to figure out cause, but what is it exactly that you're doing differently? And just thinking about it in this way really helps. And the other panelists mentioned something that raised something in my mind, which is that, yeah, I mean, I think one of the key strengths of doing this real world evidence is that the data are there. You don't need to enroll another clinical trial to get there. And that's a huge reason that we want to be able to leverage that data better to see if we can generate some causal inferences from it. So I think that that's important. But I think there's another aspect of that, which is doing genomic analyses, particularly doing multi-omic analysis. Now we're talking about single cell analysis, is also really cost prohibitive, right? The costs are going down, but if we're going to do them at scale, we're going to do them to the right sequencing depth and all of these kinds of things, it ends up being very expensive. And so there's also a motivation, not only sort of in the real world evidence to sort of use the data that we have, try to get as much causal inference out of it as we can, but in planning to do, let's say, future experiments, future reporter assays, or whatever else you're doing to sort of demonstrate molecular causation, it behooves to use all of the data, the multi-omic data that we have right now, in a way so that we can get better causal signals, so that when we move to testing them, we're testing, 1,000 candidates or 500 hits rather than 10,000 or 20,000, and then we have greater faith that it's the right 1,000, right? Not that just we did some multiple testing and we got rid of a bunch, but still the 1,000 we ended up with aren't great. And so I think there's a lot of these like levels of how causal inference and how causal inference from observational data is really going to help us, save money, save time, that sort of thing.
That's awesome. Yeah. I must admit that they weren't responses that I was expecting to hear, but I think that's a super interesting way of thinking about it in terms of being able to make it. I hadn't appreciated the dimension that it was mainly on a cost prohibitive nature. In our own work, we try some of these like EDA methods, etc., that kind of try and say that you can see some sort of causal direction from things. And we struggle, if I'm honest. I think kind of to go full circle to the topic that you started with, John, as well. Something along the lines of you need domain experts to come in to make this really work with observational data. I think kind of whilst we're on the subject of data, I think kind of one question we had was that the availability of patient data varies across regions. I think as Nancy Vo was introducing, this is probably one of the reasons why the 1,000 Vietnamese genomes project exists. And this could be, I guess, due to cost, which is not a dimension I must admit I'd thought of beforehand. But I was actually thinking more along the lines of like privacy concerns and data availability. Essentially, definitely impacts the applicability of our AI. Do you have any thoughts that you could share, more generally about the situation in Southeast Asia region? How, in your experiences, is this representative across the world? Are we ahead? Are we behind? And maybe what would you wish was different, if anything?
Maybe I can have a quick comment here. So I worked for 13 years doing clinical trial in Europe and the US, and over there, it's even more expensive than Southeast Asia, not only because of privacy, but also the cost. You cannot enroll people with the right pathology, and sometimes there are only 5 people with the right pathology you can enroll in a month. So people often got to go to countries like China, Southeast Asia to enroll the right people. So cost, variability, the data, especially for pathology and body type and origin, are very important. And we are seeing, to Dr. Nam and Professor John's point, right? Having the variability of data, having projects like 1,000 genomes are really important to contribute to the richness and variability of those data sets. It took us and Dr. Nam's project a few months to select the right individual with the right phenotypes to collect the data set, so we can collect all the data for those people. So at Bean Big Data, we also understand the challenge, and we are starting to open source some of our data sets. For example, the 1,000 genome data set is one example. We also have another example when we open source 29,000 X-ray data sets, which is carefully curated, well labeled by all the top-notch radiologists in Vietnam. And all of those data have patient consent that can be used to share across all the selected research partners. So I hope those initiatives would have us like bridge the gap with lacking the variability and the richness of data for the AI models.
Yeah.
Cool. Thank you.
I may add something. So one thing I am thinking for a long time, and even now I am working on that, is building some network between Vietnam and some Asian countries, Southeast Asian country, and some other developed country. So based on those research network and based on some collaborative project, we can bring people together and then to get data in a proper way. Because in Vietnam, it's less costly than in as a developed country to get sample and to get patient sample or something. But but based on those network, based on those collaborative projects, we can grow data. People have motivation. Vietnam government or some other Southeast Asian government, for example, they have more motivation to grow data, to produce more data for the genomic project and for some other project as well. So that's the thing that I am thinking about.
Fantastic. Well, I'm afraid that I think we've hit time, and we will need to hand over to the student competition very shortly. But thank you so much, everybody. Thank you, John. Thank you, Namsivo. Thank you, Nguyen, for joining us today. Yeah, thank you for your invited talks to start with, and thank you for the discussion. It's been very insightful. Thank you. And I think without further ado, I will hand back over to Tran to introduce the final session for today.
Thank you so much.
Thanks for having us.
Yeah,
Bye-bye.
Thank you.
Thank you.